Good morning, everybody. Can you hear me? Good. Welcome. Thank you for being here. I want to make an announcement first, which is that we have a French team recording this event. And so if anyone doesn't want to be recorded, please raise your hand. Okay, everyone's okay with that? Okay, great. So I'm Stephanie Seneff. I'm a senior research scientist at MIT. I've been doing research recently for the past 10 years or so on autism, focused on autism, looking for environmental toxins that might be causing it. And it was really about 3.5 years ago that I really feel like I figured out the answer, the key answer in autism, which is glyphosate. Now, how many of you know what glyphosate is? Pretty much everybody. That's good. It wasn't a household word a few years ago, so that's good news. And so it's Roundup, right? You can go buy it and kill your dandelions. Recently, last December, Anthony Samsel, who's my collaborator, called me up and he said, you know, I think glycine and glyphosate is substituting for glycine by mistake in protein synthesis. And I was like, Anthony, that can't be true. I don't think that can happen. He said, no, take a look. I think it is. Anthony's a really smart chemist, and he really understands chemistry intuitively. I thought it couldn't happen because because the nitrogen had extra stuff on it, it wouldn't work to hook up into the chain. But I think I was wrong. And most people think it can't work. So Anthony and I have a challenge, which is to find a way to get other chemists to believe that it's right and to get people to do the experiments that prove that it's right. But I think the evidence is overwhelming. The circumstantial evidence is overwhelming. I'm here today to tell you some of the things that we found. Once he told me that, I started looking. We found enormous amounts of evidence that can explain why glyphosate is correlated with so many diseases that are going up alarmingly today in our society and making us all sick. This single feature of in getting into proteins by mistake can totally explain all of these correlations. So this is the title, Glyphosate Pretending to be Glycine, Devastating Consequences. First a quote, in a time of universal deceit, telling the truth is a revolutionary act. Okay, here's an outline. I'm going to start with a brief review. I've talked about glyphosate before in this, at this conference. I don't know how many of you have seen my previous talks or maybe on the web some stuff. I have a lot of material on glyphosate. So I'm just going to very briefly review other material that I've covered before. And then I'm going to go straight into my main topic, which is this idea of glyphosate inserting into peptides by mistake. And you can see here a list of, these are only some of the diseases that you can totally explain by this concept. Neurological diseases, neural tube defects, diabetes, obesity, adrenal insufficiency, impaired blood flow, kidney disease, liver disease, hyperkeratosis. So all those things we're going to show you specifically how you can explain them by glyphosate inserting into proteins by mistake. And finally, I'll discuss several slides on how to protect yourself from glyphosate and how to stay healthy. So here we go with the review. This is the new childhood in America. Overweight, learning disabilities, asthma, ADHD, eczema, food allergies, all kinds of food allergies, seizures, and of course autism. 1 in 88 in this slide, now it's 1 in 68. This is a really scary number, and I do not understand why the US government isn't dropping everything else to figure out why this is happening. So we should be concerned. I was concerned. I looked hard for 5 years at all the different toxins I could think of, and none of them worked. I knew there was something more going on in autism that I could not explain by the toxins I was looking at. This was when I was very lucky to have heard a talk by Professor Don Huber in Indianapolis, 2-hour presentation on glyphosate. He is a retired professor from Purdue, an expert in plant physiology, plant pathology, and he completely convinced me in those 2 hours that I had found my answer. And I immediately dropped everything else I was doing and just obsessed on learning about glyphosate. And I've been richly rewarded in terms of gaining understanding of how human physiology works. Glyphosate is a great probe to understand how it works and why it breaks when it's exposed to this very toxic molecule. So Roundup, glyphosate, this is the molecule. It's actually quite small. And here's the plot that makes me stay awake at night. This is autism prevalence in 6-year-olds in America according to the school system. And the blue boxes, and this is over time, and the beginning of the plot here is 1995, the first blue box. On the right is 2011. So over that time period, autism rose dramatically in first grade. And at the same time, the use of glyphosate on core crops rose with exactly the same curve. And this is the use over the previous 4 years. So this is looking at the child's life from the age of 2 to the age of 6, and their exposure, and that gives you a 0.9972 correlation coefficient between those two curves. 1.0 is the best you can be. That's basically coinciding. So is glyphosate toxic? Monsanto says no, that's why it's such a fantastic herbicide. It's harmless to humans, we don't have the pathway. It's a nice story. Shikimate pathway in the plants, in the gut microbes, very, very important to our health because the microbes use that pathway to make essential nutrients that we can't make. And these are precursors to serotonin, melatonin, the skin tanning agent melanin, folate, all these really, really important molecules that we depend on our microbes for that they can't make because glyphosate is disrupting that pathway. There's other ingredients in Roundup. Seralini's work in France has shown that 1,000-fold greater toxicity, acute toxicity, for Roundup compared to glyphosate by itself. And then the really interesting thing to me is the insidious effects, and this is where the substitution into protein comes into play. Glyphosate accumulates slowly over time in your tissues, and I believe it's accumulating attached to and integrated in your proteins. And that is really, really scary because it's extremely hard to get it out once it's in that form. Most of the studies that they've done are too short. You don't pick up on these long-term cumulative insidious effects. So here's some of the main toxic effects that we've talked about, and Anthony and I talked about this in our first paper back in 2013. It interferes with the cytochrome P450 enzymes in the liver, which are really important for lots of reasons. It chelates minerals, so it makes them unavailable. Iron, cobalt, manganese, zinc. These are really important for enzyme function. It interferes with the synthesis of these aromatic amino acids, which are the products of the shikimate pathway, as well as methionine, which is the core protein, core amino acid to supply sulfur, so this organic sulfur source. And this is going to give you shortages in neurotransmitters, shortages in folate, and lots of other problems. It also disrupts both sulfate synthesis and sulfate transport. Sulfate is where I started because I identified sulfate deficiency as a key factor in autism. I'm not the only one who's noticed that, and I believe that is true, and I believe glyphosate is causing that. So here's some of the mechanisms that are associated with autism. It disrupts the gut microbes. Children with autism suffer lots of digestive issues. Sulfur metabolism, glutathione is a sulfur-containing metabolite, very important as an antioxidant. Methylation pathways, which are well known to be affected in autism. Metal chelation, especially manganese. Manganese deficiency leads to a lot of things that are associated with autism, like mitochondrial function and glutamate toxicity in the brain. It inhibits the pituitary release of thyroid-stimulating hormone, which is going to affect the thyroid and have you underproduce thyroid hormone. And moms who have hypothyroidism have a 4-fold increased risk of producing an autistic child. So now this is a Newsweek article from 2016. Nice to see this in the mainstream. Glyphosate is the most used chemical ever on agriculture. Not just herbicide, but chemical period. Fungicide, insecticide. And so this is why something going up this fast and being this pervasive in the environment is a really good candidate to be causing the autism epidemic. So it's increased 50-fold since 1996, which was when the GMO Roundup Ready crops were first introduced. And these Roundup Ready crops are like corn, soy, canola, sugar beets. These are core crops of the processed food industry. So today, 50 times more is allowed on corn than was allowed in 1996. And half the American farmers are now facing all kinds of resistant weeds, which is causing them every year to use more and more glyphosate. And what's happening finally is that they're realizing glyphosate can't work anymore, so now they're going back and adding in these other chemicals, such as 2,4-D and dicamba, which are considered by most to be far more toxic than glyphosate. These are going back in now on top of the glyphosate because of the glyphosate-resistant weeds. This is a paper by Nancy Swanson. She and I have collaborated on papers since this. This one is very interesting with all kinds of plots. Genetically engineered crops, glyphosate, and the deterioration of health in the United States of America is the title. And she's collaborated, she's produced all these plots from looking at the US government data that's available online. And she's got over 30 figures in that paper showing all these remarkable correlations. Here's just 4 examples from the paper. On the top left, thyroid cancer. The yellow boxes are the thyroid cancer rates, and the red is the glyphosate, and the blue is the GMO Roundup Ready crops. You can see the glyphosate almost perfectly matches with thyroid cancer. Diabetes is also a good match on the right. End-stage renal disease, a lot of people are having problems with kidney failure. That's very well matched. And then bladder cancer is another example on the right. So you can see cancers, various metabolic diseases. She has a bunch more in her paper. Okay, so now we get into my core topic, glyphosate insertion into peptides. I'm not sure how many of you understand what I mean yet, but I'm hoping you've had enough biology to understand this. It's really not very difficult to understand. So in biology, you have the DNA code. It's 4 letters, and you make a 3-letter pattern to produce a symbol of a particular amino acid. There's about 20 amino acids. Those are the coding amino acids. They're absolutely essential. Every one of them is very important for your biology. And they're put together like beads on a string to produce all the proteins. It's quite fascinating. So the DNA code specifies what protein to make, and you make the protein according to code, and then that protein folds up and does really interesting things. The proteins are the real workhorses of the body. You know, the enzymes, they're the channels that allow ions to flow through membranes, things that receive receptors that respond to neurotransmitter signals, all kinds of things. Those are all proteins, and they're all coded by this DNA code. So when you see the code for glycine, GGC, in producing a protein, the machinery that's going to make this protein is attaching these amino acids together. It's got a proline, it's got an alanine, it's looking for the next one. It's looking for glycine. It sees this molecule over here that looks a lot like glycine, because glyphosate is glycine. It just has something else attached to the nitrogen on the glycine molecule. So then it matches, and it goes in and puts glyphosate in place instead of glycine. That's the simple idea that Anthony had, and I believe it's true, and I'm hoping you will believe it's true by the end of this talk. So here's just a schematic of an active site in a typical protein. Many of the proteins have this kind of property where they form a kind of cavity that specifically fits a particular molecule that's going to be the substrate to carry out the reaction. And so you've got this thing in this active site, and it turns out for this particular protein, the glycine is at the active site and absolutely essential for the functioning of that protein. So if you substitute glyphosate for glycine instead, what happens, you've got your glycine right there, and then you've got this extra thing stuck onto it, this thing called methylphosphonyl group, which is attached to the nitrogen and which goes into the active site and it shoves the substrate out of the way. So the substrate won't fit anymore. So for example, in cytochrome C oxidase, the substrate is superoxide. It's glyphosate sticking inside that hole where superoxide is supposed to go. Superoxide can't go in. The molecule can't work. And cytochrome C oxidase is in the mitochondria, it's crucial for producing ATP from glucose. Very, very important for generating the energy for the cell. If it's not working properly, the superoxide is sliding out and going all over the place and causing damage to the surrounding tissues. And meanwhile, this enzyme is not working. So that's really bad for the mitochondria, and autistic kids have a lot of issues with mitochondrial disorder. This is also, by the way, what I think is happening with the enzyme that glyphosate disrupts in the shikimate pathway. That enzyme actually has a conserved glycine at the active site, and in fact it's been shown that glyphosate displaces the substrate, and that's how it works to disrupt this enzyme. In fact, E. coli have come up with a smart design to replace the glycine with alanine. Alanine just has the C here in this picture and doesn't have that P part with the O's. It just has the C, a little bit of extra stuff. It makes the enzyme much less efficient, but it protects it from glyphosate. And the reason why it protects is because the code is for alanine now instead of for glycine, so it doesn't make that mistake. Very clear to me. That makes very good sense as to how it's causing the problem that it causes in the shikimate pathway. So now we have all these predicted consequences. I started rummaging through the research literature, looking for proteins that have essential glycines anywhere in that protein, and then saying, what does that protein do and what disease would that affect? And then you say, oh my God, it's all the things that are going up in step with glyphosate usage on core crops. So here are some of them, and I'm going to go into more detail on exactly how some of these are occurring. Neurological diseases, neural tube defects, celiac disease. Collagen is loaded with glycine. Collagen is in your joints, in your bones. So when you don't have enough collagen, you're going to have rheumatoid arthritis. When your collagen isn't working properly, you're going to have rheumatoid arthritis, which we have an epidemic in today, and also osteoarthritis. Steatohepatitis, which is fatty liver disease, obesity, adrenal insufficiency, impaired iron homeostasis, which is going to lead to kidney failure, insulin resistance and diabetes, impaired sulfate synthesis and sulfate transport, and cancer. So this is quite a list. And all of these things are going up today in our society. Here's some evidence from a very recent paper from 2016. I was delighted to see this paper. And they use some fancy new technology to quantify all the changes that are taking place in protein expression in the microbes that hang out in the rhizosphere. The rhizosphere, here's pictured on the right, is sort of like the plant's gut. So this is their gut, the plant's gut microbiome that's being affected by glyphosate. And one of the things they found was a significant increase in the production of enzymes concerned with both protein synthesis and protein degradation. So what this means is that proteins are being made and they're broken and they're being disassembled and remade. And this is how protein synthesis works. I was fascinated to discover that the making DNA is much more accurate than the process of making proteins. The system is designed to just make mistakes. It gets in a hurry, makes mistakes, doesn't worry about it. And then it corrects the mistakes after it sees that the protein didn't fold correctly. Because many times those mistakes are okay and it just lets it go. And it can find, because the protein is folded correctly and it's working properly, that it's okay. If it isn't, it marks the protein for deletion. It basically says, okay, get rid of this one, get rid of that one, they didn't work. It disassembles a protein and builds it again. That's how the system works. With glyphosate in the loop, you've got to do that more because you're making a lot more proteins that aren't working properly. And furthermore, glyphosate makes it hard to break the protein down. So it ends up sticking around and causing trouble. Things like amyloid beta plaque. So I found out that there are actually many other examples of this kind of thing. For example, in Guam, there was an epidemic in ALS following World War II, and it was studied, and they found out, a very fascinating story, that it was due to exposure to a toxin, a natural toxin that's made by these cycads, which these people were eating. And I suspect that the chemicals from the war caused the cycads to make more of this toxin. This toxin is called BMAA. It's a non-coding amino acid analog of serine, just like glyphosate is a non-coding amino acid analog of glycine. And so what this causes is a defective glutamate transporter, which has been linked to ALS, and that transporter has essential serines in its molecule. So this is just the kind of pattern you look for. Look for essential glycine, find out what happens. They've even done studies where they replace the glycine with something else and find out that this protein doesn't work. They don't think to replace it with glyphosate, but they should be because that's what's going on. Here's another example, multiple sclerosis in sugar beets. Another really fascinating topic that I wasn't aware of before. Sugar beets contain something called A's, which is an analog of protein, proline. Proline is another one of the amino acids. And there's a remarkable correlation between proximity to sugar beets and multiple sclerosis. And so the reason they suspect is because of myelin basic protein, which contains a cluster of prolines that are absolutely essential. And if they get substituted by A's, the molecule doesn't work, and then you get into problems with your myelin. And this, it's all around the world, in Japan and in the United States, in the areas where they make sugar beets, in Europe, you find high rates of multiple sclerosis near sugar beet fields. So now here's a whole bunch more. So I was going through finding things, conserved glycines. On the left, you have specific enzymes that have places where they have one or more absolutely essential glycines. And then you say, if those glycines are displaced by something else, that protein doesn't work, what do you get? And on the right are the diseases that you will get from these. And this is how you can see obesity, diabetes, Alzheimer's, cancer, kidney failure, adrenal insufficiency, mitochondrial disease, Parkinson's disease, and ALS. All of these things are going up exactly in step with glyphosate usage on core crops. And so I think glyphosate is going into those proteins, causing those problems, causing those diseases. Okay, so we're going to go into a little more detail about some of these diseases, starting with glyphosate in vaccines. This is a topic that has me greatly concerned. We haven't proven this, it's only a theory, so we're looking into it. But I suspect some of the vaccines are contaminated with glyphosate. The reason why I suspect that is because the vaccines are, the live virus is grown on gelatin. The gelatin is derived from the ligaments of pigs. The pigs are fed a heavy dose of GMO Roundup ready corn and soy feed. There's no doubt the pigs are being exposed to lots of glyphosate. And the gelatin comes from collagen, and collagen is loaded with glycine. It has 1/4 of the residues in collagen is glycine. So you can expect glyphosate to substitute for many of those glycines in the collagen, that then the virus is grown on the glyphosate. The virus puts the glyphosate into its own proteins and makes them toxic. So I'll have more to say about this topic on Friday, but I've just got a couple of slides here today. The other thing is that gelatin contains glutamate as well as glycine. And so glutamate is actually a known neurotoxic, excitatory neurotoxin. And glyphosate exposure through the food is going to give you a leaky gut barrier, followed by a leaky brain barrier, which is going to allow these toxins in all the vaccines, the aluminum, the mercury, the glutamate, more access, easy access to the brain. And so the exposure from the food is going to set the child up to fail when the child gets the vaccine. And so, and meanwhile, you might also have glyphosate in the actual proteins that you're supposed to develop an antibody reaction to in the bug, in the microbe, such as the measles virus. You can have, if you have glyphosate in those proteins, they're going to be hard to break down, and your body could end up developing too many antibodies, and especially in the brain. The brain's immune cells could develop antibodies, which could then lead to autoimmune disease. So glyphosate stimulation of the nimbus receptors is going to cause neuronal burnout, and that's actually been shown in papers that have been done on mice, where they exposed these mice to an acute exposure to glyphosate of these mice pups. And they showed that the NMDA receptors were overexcited, calcium channels got on fire, oxidative stress, neural cell death, increased glutamate release, which is going to then cause this excitotoxic reaction, depleted their glutathione, which is an important antioxidant, and increased the damage to the lipids in the brain. So here are 4 neurological diseases that are on the rise today: Alzheimer's, Parkinson's, ALS, and then the prion diseases, such as mad cow. And all 4 of them, each of them, you can find at least one paper that talks about a protein associated with that disease that misfolds. And this is, for example, the amyloid beta plaque, which some of you probably know about in association with Alzheimer's, but there's also one for Parkinson's, one for ALS, and then the prion proteins for the prion diseases. In all those cases, all those proteins have essential glycines, and in fact, even more remarkably, the glycines have been targeted as the suspected problem in the protein. They don't understand why the glycine is a problem, but they think it is, because they've been able to narrow it down to specific peptides that contain highly conserved glycines. It's really, really remarkable that all of these diseases have that property. And I think it's just that the glycine is getting substituted by glyphosate. The protein misfolds, and then you can't clear it because the glyphosate interferes with your ability to break it down. And glyphosate will also interfere with your ability to break it down because of the sulfate depletion, because sulfate is essential in order to be able to recycle cellular debris. So that's 2 things working together to cause harm. So in all cases, it's a rare soluble peptide rather than the precipitated plaque that's causing the problem. And glyphosate increases the protein solubility, so you would expect this to happen. Okay, deaths from senile dementia, just to show you one example, perfect match with glyphosate usage on corn and soy crops. This is going from 1979. Glyphosate was introduced in 1974, 1975. So this plot goes from 1979 up to 2009. And 1997 is around the time frame when they introduced the GMO Roundup Ready crops, and you can see an accelerated growth in Alzheimer's, in dementia at that point. Now, GABA is another interesting thing, and there's a brand new paper out, 2016. I was excited to see linking GABA receptor deficiency to autism and a weakened response. GABA is an inhibitory neurotransmitter, so it calms the brain down. So if it's not working, then the brain doesn't get calmed down. The child gets overexcited. GABA receptor has a conserved glycine at the entrance to the first membrane-spanning domain that's essential for its function. So if that's replaced by glyphosate, the protein won't work, the GABA receptor will not receive the GABA, and you will get overexcited brain. Okay, neural tube defects, and I'm going to talk more about this on Friday. Glyphosate and anencephaly, I've shown this slide before, I think. Yakima, Washington, they have an epidemic in anencephaly, children being born with no brain. They were concerned about it. They took a look at some pesticides. They looked at 75 different pesticides. They analyzed them. They couldn't find any correlations. Of course, they didn't look at glyphosate because glyphosate is harmless to humans. However, they were using huge amounts in exactly the same years in which this epidemic appeared. A 5% solution of glyphosate was used heavily around the irrigation ditches to control the weeds. And it was the main herbicide recommended because of its quote-unquote low toxicity. But glyphosate has actually been linked to anencephaly due to its effect on retinoic acid. And this is, I'll do this in a moment. I have this one extra slide just to show some pictures. This is a slide that was given to me by Don Huber. He's the one who first alerted me to glyphosate. And he shows here that little red arrow at 2010, huge increase in the use of glyphosate on these weeds in the waterways, coinciding with this epidemic in anencephaly. So why they can't connect these dots, I do not understand. How might glyphosate cause anencephaly? Many ways. Neural tube defects are linked to folate deficiency. Folate is a direct product of the shikimate pathway. That's going to give you impaired methylation capacity and THFR. I mean, all these things that we talk about in this conference about the folate and the methylation pathways and their defects in autism can be connected to glyphosate because of the microbes not being able to make the folate through that pathway. And they're especially sensitive to glyphosate too, so they get killed as well. It also interferes with retinoic acid metabolism, and this goes to a toxic accumulation of retinoic acid, which is linked to neural tube defects. And then it disrupts the glycine cleavage system. Glycine itself is a source of methyl groups, and so you get both folate deficiency and methyl deficiency in the presence of glyphosate. So here's a really great paper by Carrasco. It was amounts of exposure that were far below the concentrations that were used in agriculture in this paper. They exposed these tadpoles to glyphosate. They found that retinoic acid went up. Now the reason is because retinoic acid is metabolized by the cytochrome P450 enzymes in the liver, which are defective because of the glyphosate exposure. When retinoic acid is too high, you end up with things like problems with the brain, with the neurotube development, like cyclopia, which is one eye, microcephaly, various neurocrest problems, but microcephaly in particular is a close cousin of anencephaly. And microcephaly is what's going on right now in Brazil, and I have my talk on Friday will talk a lot about the microcephaly epidemic in Brazil and how there are many, many other factors besides Zika virus that are causing that. Okay, so methylfolate and one-carbon metabolism. Glycine is a key source of methyl groups for the one-carbon cycle via the glycine cleavage system. So here's a picture of a glycine molecule. Glycine is actually the tiniest amino acid, doesn't have any side groups, but this is a carbon, that black is a carbon, a methyl group actually, that's going to get yanked out of that glycine molecule and used to supply methyl to folate through this methylation pathway. So to get that methyl group out, you have to apply an enzyme called glycine decarboxylase, which is essential, and that enzyme depends on a glycine-rich region in order to maintain the shape and flexibility of the molecule to be able to do its job. Okay, diabetes, obesity, and adrenal insufficiency. Here's a rather complicated-looking picture. The thing across the top is the cell membrane. The outside world is on the upper part. Inside is the cell. All these different things are going on in the cell. I really want you to focus on just three things in this picture, which is this hormone-sensitive lipase, also called triacylglycerol lipase. That's a key hormone, a key enzyme, protein, in fat cells. And it responds to hormones. The hormones could be things like adrenaline. And in response to the hormones, it releases, it orchestrates this stuff on the right that causes the release of fats from the fat cell. Those fats are going to be released in order to supply energy for sort of the flight or fight response. So you need to be able to have energy. You metabolize the fats that are being released from the fat cells. But if this protein is broken, you won't do that. So that's going to be two consequences. One is that you won't have energy. You'll be drained, and exhausted. But two is that your fat will pile up in your fat cells and you will gain weight. And of course, every time any country starts to switch over to a Western diet, people start gaining weight, and they just say, well, we're just sloths, we're lazy, we're not exercising enough. The fact is we're being poisoned, and we're being poisoned in such a way that we cannot get the fat out of our fat cells. So that's why we gain weight. It's not our fault. Hormone-sensitive lipase also has two important roles. One is to let the fats out of the fat cells, but the other one is to let cholesterol into the pathway that's going to produce a huge number of really important hormones, such as adrenaline, a huge bunch of hormones in the adrenal glands from cholesterol. And that process is going to be disrupted if this enzyme is broken. So it turns out this enzyme has lots of dependencies on glycine. It contains something called an HGGG motif, which is this oxyanion hole. GGG means 3 glycines in a row. It's a member of the class of molecules called serine proteases, and these have all kinds of important roles, and they're all going to be affected, not just this one, but all the serine proteases. They contain a GXSXG motif, 2 Gs, one on each end, that's essential for their activity. So anytime you get glyphosate in these proteins, they're not going to work properly. And impaired hormone-sensitive lipases has been linked to obesity, atherosclerosis, and type 2 diabetes. So here's just a plot of death due to obesity, which is, of course, something rare, but you can see that death due to obesity is not as rare as it used to be, and in fact, it's also going up exactly in step with glyphosate usage and in general obesity in the population. I've seen a plot where you see 1975, we were getting fat slowly up to 1975, and then it hits a corner. We start getting fat much faster right when glyphosate was introduced into the food chain. Adrenal insufficiency is also an epidemic, and it has a lot of nasty symptoms. Roundup has actually been shown in experiments to cause adrenal insufficiency, impaired adrenal hormone synthesis, and I think one of the ways it does it is through impairing this particular protein that I just talked about. So the insulin receptor in diabetes, this is also a very straightforward story. Glucagon release is regulated by insulin receptors. So in the liver stores glucagon, which is a kind of storage form of glucose, and so it will be able to release that and turn it into glucose in a situation where there's low blood sugar. And so this can keep the blood sugar levels high enough in your blood. But when these receptors are dysfunctional, what happens is the liver releases too much glucose, and you end up with hyperglycemia, which is a step towards diabetes, excess blood sugar. And these receptors have 8 repeats, so 8 in a row, of a glycine-centered motif, which determines the three-dimensional structure of the molecule, makes it work properly. And it also has an ATP binding site that contains a GXGXXG motif, 3 glycines in that, that is essential. So any of those glycines messed up, this protein is not going to work, and that's going to directly lead to diabetes. And so here's a plot of diabetes and glyphosate. Again, the yellow boxes are the diabetes, the red is the glyphosate, and the blue is the GMO Roundup Ready crops, which you can see how well the glyphosate tracks the GMO Roundup Ready crops as well. Okay. Okay, celiac disease. So, huge epidemic in celiac disease these days. You go to the grocery store, it's just, you expect to see a huge section selling gluten-free items. 10 years ago, that didn't exist. I didn't even know the word celiac disease. It's just amazing how this has just come out of nowhere. And I might add just the more general problem of food intolerances. When you look at the allergies that we're facing, the peanut allergies, the soy allergies, the casein allergies from the milk, these are allergies to proteins, and every one of those proteins is likely to be contaminated with glyphosate. And so I think more generally, peanuts are sprayed with glyphosate right before the harvest, for example. And so is wheat. And so I think we're just getting glyphosate into the proteins, and as a consequence, the proteins can't be broken down, and then the body develops an immune reaction to the proteins, and then it gets these sensitivities that you can't eat those foods. Anthony Sampson and I wrote an entire paper on celiac disease, and we missed this point because we didn't, what I'm going to talk about in a moment here, because we didn't know about the idea that glyphosate might be substituting for glycine in protein synthesis. So it turns out that gluten contains a lot of prolines. Proline is one of those amino acids, remember, that are essential. Gluten contains a lot of prolines, and proline is actually difficult to break down. Now, the reason why proline is difficult is actually interesting. It's because it has something attached to the nitrogen. It has an extra carbon attached to the nitrogen, which is what glyphosate has. And so when I saw that, I hadn't realized that about proline. It's the only amino acid besides glyphosate that has an extra carbon attached to the nitrogen. But that does not stop it from going into the protein chain. And that means that my original thought that glyphosate couldn't be going into the chain was wrong, because proline is perfectly happy to do it, and it has the same model. And once I saw that, then I was much more comfortable with the idea that Anthony had, that glyphosate is getting into proteins. So proline, because of this extra carbon, is hard to break down. That means glyphosate is going to be hard to break down too, and in fact it's been shown that glyphosate, in fact they've even made chains of glycines with different things hooked onto the nitrogen, not just the methylphosphonol of glyphosate, but other molecules that have all kinds of stuff hooked onto the nitrogen. They can put those things together as chains, just like peptide chains. They call them peptoid chains. There's a whole research literature on this topic. And they're excited about these things because they can put onto the nitrogen various molecules that might have interesting biological effects that they could use as drugs. And the reason why they like it is because this thing is really hard to break down. So it sticks around a long time. I find that really scary. And that also means that these proteins that have this glyphosate in them are going to have that same problem. So prolylaminopeptidase is an enzyme that specializes in breaking the bonds of proline, the difficult amino acid to break down. And so, first of all, it depends on manganese as a catalyst, and manganese is chelated by glyphosate and made unavailable. So that's going to be one problem. This is in the gut, so you're going to have the glyphosate there affecting this protein in the gut. But secondly, it contains a highly conserved GXSXGG motif and 2 other regions with conserved glycines. So 3 different parts of this protein have highly conserved glycines. Any of those can be susceptible to glyphosate substitution. And so if this molecule doesn't work, then you can't break down the gluten. And on top of that, if it's got glyphosate in it, it's going to be even harder to break down. So on both sides of that fence, you're going to have trouble breaking down the gluten, and that's why you're going to end up with, especially with your leaky gut, you're going to get the gluten out into your system, and it's going to cause a reaction. And so, but of course, malabsorption due to celiac disease can lead to nutritional deficiencies because the celiac messes up your body's ability to absorb various nutrients, vitamins and things like that. And that can cause symptoms of autism. Okay, celiac disease. This is a picture from the paper that Anthony and I wrote on gluten intolerance and glyphosate. And you can see here that the yellow boxes are celiac disease, and the green line is glyphosate usage on wheat, not corn and soy, but wheat. And you can see quite a good match here between the rise in using glyphosate as a desiccant on the wheat right before the harvest and celiac disease. Okay, impaired blood flow, endothelial nitric oxide synthase, probably my most favorite enzyme. Really fascinating molecule, and I've written a lot about it. My colleagues and I have a theory about enos that's quite I really believe is true, and we need validation. We haven't proven it. It's one of these things that makes a lot of sense. Enos is famous for being able to make nitric oxide, and nitric oxide relaxes the blood flow, so it's really important in the vasculature. It's released by the endothelial cells, and that allows the blood to flow. But enos also has a pathological state in which it releases superoxide instead, and this is called enos uncoupling. There's lots of papers about this. Enos is releasing superoxide instead of nitric oxide. When you mix nitric oxide with superoxide, you get something called peroxynitrite, which is really toxic. And some people have thought that peroxynitrite is behind many of the modern diseases that we're facing because it's so toxic. So you don't want to be mixing superoxide and nitric oxide, but enos is what's called a moonlighting enzyme. It's a neat word, moonlighting. It actually switches back and forth between 2 activities, one of which is to make the nitric oxide, but the other one is to make sulfate. This is our theory, and we have papers on this. When the enos is attached to the membrane, it makes sulfate, and it uses superoxide to make that sulfate. But if it's substituted by glyphosate, it can disrupt its ability, just like the cytochrome C oxidase. It can end up spilling out the superoxide and causing trouble, and not actually making the sulfate. Double hit. Very, very serious consequences of that. So enos turns out to depend on conserved glycines both for dimer formation and for binding to the membrane. So enos operates as 2 enos molecules together with a zinc atom sitting inside a cavity, and the zinc atom draws in the superoxide, I believe, to make the sulfate. But so if you can't make the dimer, you can't make the sulfate, and the superoxide is just going to kind of go wild. And furthermore, enos only does this when it's attached to the membrane. So if the other piece which has glycines that are supposed to attach, there's a terminal glycine on enos that makes it attached to the membrane. If you replace that glycine with anything else, it won't work. They've done that in the experiments. So you replace it with glyphosate, it can't attach to the membrane. If it can't attach to the membrane, it can't make sulfate. And that's why you end up with a massive problem with sulfate deficiency systemically. So I think this is really exciting. I was just delighted. Enos is also, by the way, a CYP enzyme. It's a cytochrome P450 enzyme, so it's going to also be affected by glyphosate through that pathway. So it is really getting clobbered by glyphosate, and I think that is a huge issue in our failing health today. Impaired eNOS function leads to oxidative damage in the artery wall, and of course also this peroxynitrite that I mentioned gets produced if there's nitric oxide lying around as well. So it's going to be a deficiency in nitric oxide and a deficiency in sulfate and excess collateral damage due to these superoxide radicals. Kidney disease. So the kidneys actually have a very interesting way of managing their iron, and I only learned this recently during this episode of trying to figure out all these proteins with their glycines. It's certainly the case that we have an alarming incidence of kidney failure here in this country, as well as most especially among young agricultural workers in sugarcane fields in Central America and also in rice paddies in Sri Lanka. The Sri Lankans have figured it out that glyphosate is the cause of this kidney failure, and as a consequence, they've taken action. Their government, unlike our government, was reacted and said, oh my God, and decided we're going to ban this molecule. So they do not allow glyphosate for any purpose in Sri Lanka today. It's really great that they've done that. Because it was clear to them that glyphosate was causing this kidney failure epidemic in their young agricultural workers. So this particular kind of failure that glyphosate produces is actually very specific to these things called the proximal tubules, and it causes this necrosis of those tubules. And of course, the sugarcane is sprayed with glyphosate right before harvest, and so these workers who are spraying it are the ones who are getting sick. So what is really interesting to me is how the iron is managed by the kidneys. It actually is a symbiosis between human cells and bacterial cells. And the bacteria actually produce these things called siderophores, which are able to contain the iron inside a safe capsule. This is really important because free iron is extremely toxic, and it'll react with the superoxide and hydrogen, the hydroxyl radicals, and cause all kinds of trouble. That's well known. So these siderophores are going to hold on to the iron and make it safe, deliver it to the kidney, and then the kidney has actually special enzymes. These are cross-species enzymes that say, oh yes, good, here's a siderophore provided to me very nicely by this microbe. I will take it up and use it for my iron. And that's a way to get the iron delivered to the kidney safely. So if those siderophores aren't being made, or if those receptors aren't working, the kidney is going to have iron deficiency problem, and also there's going to be an iron toxicity problem because the iron is now free rather than being hiding inside the siderophores. And that's exactly what you get. So here's a paper that talks about this really fascinating process by which bacillus, this microbe bacillus, produces these siderophores, which look like this structure here. And then they hold on to the iron and keep it safe so that it's not going to cause damage. And then this tubule has this specialized enzyme that takes up the siderophore, and then the tubules can use that as a source of iron, which is really important for their catalytic activities. And so both the siderophore and the receptor have critical conserved glycines. The siderophore has a couple of conserved glycines, and the protein that takes it up has a conserved GXW motif. So here's a paper, here's a plot from a paper that Anthony and I wrote. We didn't know about this particular piece of the story at that time, but we showed that there was almost a 7-fold increase in the incidence of glomerulonephritis over time from 1998 to 2010, which is the condition that is causing this kidney failure in these agricultural workers. And so now I feel like we can explain it through this defective iron uptake by the kidneys. Liver disease. Okay, liver cancer and glyphosate. Here's a picture of increased rate of liver cancer starting around 1996. Again, the yellow boxes are the cancer, the red is the glyphosate, and the blue is the GMO Roundup ready crops. Fatty liver disease is a strong risk factor for liver cancer, and we have an epidemic in fatty liver disease. We have a sort of non-alcoholic fatty liver disease, NAFLD, that's caused by fructose and stuff like that. We have a lot of issues with people getting fat stores, excess fat stores in their livers. This plot on the right looks complicated, but we're just going to look at a few pieces of it. These things called Mallory-Denk bodies, this is the MDB formation on the right. These things are associated with this fatty liver disease, and they are caused by this pathway that's illustrated in the rest of the picture here. KEAP1 is the protein that I'm focusing on. KEAP1 is an interesting protein which hangs on to NRF2. So you see KEAP1, NRF2. KEAP1 grabs hold of NRF2, and then it also grabs hold to the cytoskeleton, and it says, okay, NRF2, you can't go into the nucleus. It holds it there attached to the cytoskeleton. if KEAP1 lets go of NRF2, or if KEAP1 lets go of the cytoskeleton, then the NRF2 can get into the nucleus, and when it does that, it'll turn on a whole bunch of protein synthesis and cause a whole bunch of things to happen. So KEAP1 has a conserved double glycine repeat that anchors it to the cytoskeleton. If that thing is glyphosate, it won't be able to anchor, and then the NRF2 will be able to get into the nucleus and cause the release of K8, which is a keratin. And keratin then is what causes the formation of these bodies which have keratin in them. And so there's another interesting story that I want to end on before I get into how to fix the problem. Hyperkeratosis, this is a problem in us as well, I believe, and also in racehorses, for example. They're having a lot of issues that I think are connected to hyperkeratosis. I'm looking into that right now. But I want to focus on the birds at the moment. Beak deformities in the chickadees. And here's a picture of a chickadee that can't eat because its beak's so twisted around and deformed that it can't open seeds. And there's an epidemic in this problem in the chickadees, particularly in areas located near agricultural regions. Starting with the Great Lakes region in 1998, which is when they were just starting to get going with the GMO Roundup ready crops, in central Alaska, and in areas exposed to the California agricultural runoff. All these areas, they've got these chickadees that are having troubles with these beaks. Below here, I have the reference to a paper that was written in 2015, very detailed. They looked at lots of different metals, toxic metals. They looked at lots of different agricultural chemicals, big lists of stuff, couldn't find any connections. Same story, just like the anencephaly in Washington State. They didn't look at glyphosate at all and didn't find any connections. But it's easy to explain this with respect to glyphosate because of this effect that I just mentioned with KEAP1. And furthermore, these chickadees are going to be eating sunflower seeds, and the sunflower seeds are sprayed with glyphosate right before the harvest. The chickadees are a very popular bird at the bird feeders. So they're getting glyphosate from their sunflower seeds, and that's causing their beaks to have this problem. And it's also a systemic problem in their body. It's showing up elsewhere in their problem as well, this overexpression of keratin. And as I said, we have similar problems ourselves. So finally, how to safeguard yourself and your family. This is fun. I really love this. Dandelions, eat dandelions. Dandelion leaves. So Roundup is toxic. This is a paper, a nice paper that talks about these common everyday plants that are healthful. They can provide you nutritional support for fighting glyphosate. And so they talked about the toxicity of Roundup to the liver and to embryonic cells at very low doses, far below the doses in agriculture. And so they said dandelions, barberry, and burdock. They mentioned specifically those three common plants that can protect from damage, especially if you eat them before you're exposed. And this was a lovely paper on cows. These cows were getting sick, and they tested their urine for glyphosate and found out that they had glyphosate in their urine. And I guess they've developed this treatment method, which is based on activated charcoal, bentonite clay, humic and fulvic acid, which are organic matter from the soil, and sauerkraut juice, which is going to be loaded with probiotics. So these are the treatments that they administered to these cows, and they showed that their symptoms improved and that the levels of glyphosate in their urine went down following these treatments. So I think humans could also make use of treatments of this sort. These things are pretty non-toxic, and I think they're available. Good things to think about in trying to detox glyphosate. Here's some nutrients, and of course there's many more, but these are some of my favorites. That can help you, particularly help you heal the sulfate problem, because I think the sulfate problem is the key one. If you can get your sulfate levels built back up again, your brain won't be leaky anymore, your gut will heal, everything will fall into place, your blood will flow. So really, really important to get your sulfate up, but it's not easy because sulfate's hard to transport. And I talk a lot about this in some of my other talks, but today I don't have time, so I'll just mention it briefly. But some of these are sulfur suppliers, such as glutathione. Glutathione contains sulfur. And taurine is a really interesting molecule. It's an amino acid that contains a sulfonyl group, which is almost sulfate. So taurine can be a good source for sulfate. I love Epsom salt baths. You just get into the hot bathtub and you dump in some. You can just get the Epsom salts very easily at Whole Foods, for example, and dump them into the bath water and just soak for 20 minutes. The sulfate, the Epsom salts are magnesium sulfate, and magnesium is also a good thing to get. So you get naturally magnesium and sulfate soaking through your skin bypassing your gut, because lots of times people have issues with sulfur in their guts. People have sulfur-reducing microbes that have overgrown, probably because of glyphosate poisoning, and they can end up producing a lot of hydrogen sulfide gas, which can cause a lot of nasty issues. Probiotics, of course, I'm sure most people here know that. Vitamin C is a good one. Vitamin C is actually also carries sulfate. And in fact, I suspect that vitamin C facilitates the transfer of sulfate onto the artery wall to help to make the blood work properly. Really, really nice. Vitamin C is very good. Of course, garlic I love. Eat as much garlic as you possibly can. That's a good source of sulfane sulfur, which is a really nice way to get your sulfur. Methyl tetrahydrofolate, I want to emphasize that's not folic acid, and I do not recommend folic acid. I've talked about that in previous talks. Folic acid is oxidized and it's missing the methyl group. So what it does is it works your liver real hard to try to fix it and can deplete your supplies in your liver. So you want to take methyl tetrahydrofolate if you're going to take folate. And cobalamin is B12, and that's another one that's actually crucial for sulfate synthesis and for some other things as well. So I think that's curcumin. Curcumin is what people often believe that's why the people in India have low rates of Alzheimer's, because of curcumin, and curcumin is also a really good sulfate transporter. Get grounded. So again, in other talks, I've talked about the electricity in the body and how essential it is and how the sulfates are important for making the electricity work. Sulfates have negative charge, but you can get negative charge right out of the ground. The Earth is a giant negatively charged battery, so you just get the electrons flowing right into your body and helping you to regenerate the negative charge. Walking barefoot in the water at the beach is one of the best things you can do, especially on a sunny day, because the sunlight is also important. This is a picture of my kitchen table with various organic products. We don't buy anything if it's not organic anymore for our kitchen. When we eat out, we cheat because we can't find organic restaurants. I'm hoping eventually you'll have the organic choice, or even you'll have the organic restaurant. These are coming, and I'm looking forward to that, because then we don't eat out very much because of this. And when I travel, I bring stuff to eat in my suitcase, which just seems really silly, but I have to because I can't afford to get too much poison, I try to keep it low. Eat foods containing manganese. Some of these are wonderful foods. The oysters, any of the seafoods are really, really healthy. Seeds and beans, and of course the greens, the dark green vegetables, these are all really good. Tea is a good source of manganese, and fish is also. And tofu is actually a good source of manganese, but I don't recommend tofu unless you can be sure that it's organic. And bread too, same problem. It has to be organic if you're going to eat bread. These are some foods that contain sulfur, and sulfur is super, super important. Cabbage, all the cruciferous vegetables, broccoli, cabbage, Brussels sprouts, cauliflower. Eggs are a great source of lots of things. I really encourage eating, especially the egg yolk, eating lots of eggs. And healthy eggs, of course. Scallops, all the seafoods, clams, lobster, crabs, scallops, those are all really good for you. And liver, if you could get it organic. Liver will be toxic if you can't. And chicken and nuts, and of course garlic and onions also, they're good sources of sulfur. So, in summary, glyphosate contamination in our food supply is a serious threat to health. Toxic mechanisms include mineral chelation, disruption of CYP enzymes, and disruption of gut microbes. I believe glyphosate is the most important factor in the autism epidemic. Glyphosate may erroneously replace glycine during protein synthesis, and if it does, this explains why we have all of these correlations between glyphosate and a whole bunch of diseases that are on the rise today, including diabetes, obesity, Alzheimer's, other neurological diseases, liver and kidney disease, vascular diseases, celiac disease, etc. Eating an organic diet rich in sulfur and manganese and probiotics can help protect from glyphosate poisoning. Thank you. So do we have time for questions? Or? Yeah. Oh, you can go ahead. Yeah. Oh, yeah. Go ahead. And then we'll get you next.
Of the sulfur in the urine.
Yes.
So the kidney cannot hold it, and if it's low in the serum, you lose it. Yes.
Yes.
And the only thing you can do, you can take Epsom baths or something like that.
Right.
But how does it go into your picture? With the you said the enzyme is also. The enzymes is making sulfate. And
Yes, the enzyme enos, which makes sulfate, is defective, I believe, as a consequence of glyphosate poisoning.
they're more deficient. So
And also sulfotransferases, I didn't mention it, but they also have conserved glycines. These are the things that transport sulfate from one molecule to another, which are really important because sulfate is moved around a lot. Sulfate free in the blood is dangerous because it'll gel the blood. So you have to have these carrier molecules. And there's cholesterol and all the different sterols, vitamin D, and then all these neurotransmitters and the aromatic amino acids. All of these things transport sulfate and then hand it off to the artery wall to supply the sulfate in the artery wall, which will keep it working properly to make the blood flow. So, and also sulfate flushing through the kidneys is because of vitamin D deficiency. Vitamin D protects from that. Vitamin D the CYP enzymes are needed to activate vitamin D. So we have an epidemic in vitamin D deficiency that's due to, I think, glyphosate poisoning of the CYP enzymes.
okay, thank you.
I've got over here, right? Yeah. Mm-hmm.
So I notice a big correlation with kids with autism and ADHD and sensory integration disorder with Pilar's keratosis. And could that be related? Keratosis kind of chicken skin.
Keratosis, oh wow.
Could that be related to this?
Absolutely, yeah. In fact, NRF2 is a response to oxidative damage. And you have oxidative damage because the cytochrome C oxidase isn't working because of the glyphosate. That causes the superoxide to get released. That causes NRF2 to be activated. And then on top of that, KEAP1 can't keep it out of the nucleus because it does, if its glycine gets disrupted, it can't attach to the cytoskeleton. So that's two different ways in which you get over stimulation of NRF2, which gives you excess production of keratin, which I think is causing, for example, choke in the horses, and also possibly their problems with their hooves. They have this founder. The race horses have a lot of problems that I think are due to glyphosate poisoning. I want to write a paper on that, but I haven't yet. But that's interesting. Keratosis in association with autism? Yeah, that's I should learn more about that.
Okay, I have a question. You had mentioned trying to avoid exposure through eating organic food. Has there been testing about how common it is in water? Do we need to be worrying about trying to do special purification of water? And if so, what has been found to be the most effective way of eliminating it from our kids drinking water? And kind of conversely, what are we finding is helping break it down in nature, or is there anything that is, and what's the estimated time for that?
That's a good question. The water problem I don't know very well, and I don't know if anybody in the audience knows. I would love to have the answer to that, whether the actual filtration processes that go on how the water treatment plants, do they, in fact, break down the glyphosate? I suspect in part it has to do with what microbes are growing there, because there's actually only a few microbes that are smart enough to break that CP bond. The CP bond in glyphosate is a really strong bond and hard to break. And interestingly, Pseudomonas aeruginosa can do it. And also there's a microbe called Acinetobacter, which causes like a runny nose and gets into your lung cavity. That can break down glyphosate. And so I suspect a lot of kids have runny noses today all the time, it seems like to me. And I suspect that's Acinetobacter at work detoxing the glyphosate. So in the soil, you have certain microbes that can break it down. But there's only a few. It's actually quite remarkable that not very many of the microbes are able to break that CP bond.
How would those microbes...
Zen has an answer? Oh, good. Zen has an answer on water. I should expect her to, because she's done a lot of work with water, so she'll be able to clarify that in her talk coming up next. Is that it?
Well, I was going to ask, as far as the microbes go, do you know if the ones that would help with that are often eliminated by the chlorine they put in water?
Could be, could be. But I will say the ones that are able to fight it in your body are often also pathogens. So Salmonella and Pseudomonas are both serious pathogens that have multiple antibiotic resistance. And glyphosate actually causes multiple antibiotic resistance. I haven't talked about that today, but I believe that. There's papers that have shown that. And those two microbes, because they can metabolize glyphosate, they're able to survive when the other ones are getting killed off. And that's why we have that epidemic in those issues, I think.
I'm Lorena Benarroch, clinical immunology from Venezuela. Sorry, I can see her. And you mentioned about the live vaccines increasing the antibodies. We have a big cohort where we have found very big increased amounts of IgG, 7,000, 12,000 for mumps or varicella or even measles.
Oh,
which
yes.
cannot be explained by any of the immunologists I've tried to reach out to.
Wow,
And
interesting.
maybe what you're talking about would be the way to get to the problem.
I think you're probably right.
Can you elaborate on that?
Yes, well, in fact, I'm going to talk more about the vaccines on Friday. I found it very, very interesting, and what I suspect is that in the case of the measles virus, which is the MMR vaccine, as I said, the measles is growing on glyphosate, if I'm right, with the gelatin, puts it into its proteins. There's a particular protein, measles hemagglutinin. And that protein, and autistic, there was a paper from 2002 that showed that autistic kids, they looked at 125 kids, and 60% of them had really high titers in response to the hemagglutinin. So hemagglutinin is what you actually need to respond to in order to get the antibody response that they're looking for to be vaccinated. I mean, another paper showed that hemagglutinin is the one you care about. But if you get too much hemagglutinin antibody, especially if the virus gets into the brain, and then your brain's immune system develops those antibodies, then hemagglutinin resembles, there's this peptide in hemagglutinin that resembles myelin basic protein. And so you get autoantibodies to myelin basic protein. And 90% of those kids had autoantibodies. 60% of the autistic, 90% of them, over half the autistic kids had autoantibodies to myelin basic protein, which is going to attack the myelin sheath and probably cause the autism symptoms. So to me, that seems like the most plausible explanation for autism that I have heard.
Thank you.
Thank you for your talk, Dr. Seneff. I have been studying chlordane poisoning for several years, which was a pesticide that was outlawed in the 1980s, and I had a very toxic exposure to that, which I believe contributed to my brother contracting leukemia and subsequently passing away, and both of us have severely affected autistic children.
Wow.
Now, I know chlordane is not your area of expertise, but I did just want to share with you that I do believe in my own research that people of my generation who are producing these children who have autism, it could be something going on in utero with that. And we are trying to learn ways to detox from chlordane, though there's not much information out there. And I'm wondering, and you may or may not know, but it's certainly going to be beneficial for me to use some of the therapies that you suggested for detoxing the glyphosate, which is also an issue because we used to eat a normal Western diet, which we don't do any longer.
Right.
But I wonder if just getting these pesticides out of our body by these chelating methods that you spoke about, and we also have been using homeopathy to clear ourselves of the chlordane, if that would be beneficial, as well as the fact that I think it's important for us to all think about maybe different ways that we can treat our lawns. Because
Yes.
we're being exposed by the landscapers to everybody spraying
I
on their
know.
lawns.
I know.
So is there a suggestion that you have for what we can tell our landscapers to use? I mean, nothing?
I actually
organic?
met someone just this past weekend at a festival who has a who is cutting edge organic on how to treat lawns. So I got his number, and I'm going to contact him and have him do our lawn. We have lawn service, and we absolutely refuse to let them use any of the toxic chemicals. So as a consequence, we have a bunch of weeds in our lawn. And our neighbors don't like that, but we don't care because it's just like people need to get used to the idea that it's okay to have dandelions. I mean, in fact, they're useful, you know. Don't worry about the weeds. I just think it's crazy that we think we have to have these beautiful manicured lawns, and as a consequence, we're poisoning our children. It's so stupid. Yeah, I mean, we just need to get over that, you know. And look, think of the soccer field too. I suspect the soccer field is loaded with glyphosate, and all the soccer players are getting sick from that. You know, so it's just, we need to fix it. We need to be aware that we don't need to have beautiful lawns. We just, and of course there are organic ways to, you know, vinegar and there's some things you can do, but they're not as effective, of course. But that's, you know, of course you can always pull weeds by hand, which is what I do, but that gets tedious, you know. But you just shouldn't have to obsess on the weeds, I feel. Are we?
What further studies do you want to see done to prove your
Oh,
glyphosate
yeah.
replacing glycine?
It actually shouldn't be hard. I'm trying to goad these people on to do it because they keep thinking the burden should be on us to prove that it's there. But I don't see why the burden shouldn't be on them to prove that it's not happening. You know, it's equal. And they can do it. I can picture the experiment you would do to prove this. And so I've been contacting people and saying, hey, you know, why don't you do this? I don't have a lab, so I'm not equipped to do it myself. And of course, I don't have money either, so there's some issues, and no one wants to fund something like this because, so it's going to happen eventually. I'm just going to get them, I'm going to shame them into doing it, basically, I think. I hope. Either my side or their side. Somebody's going to decide to take a look. Anthony is finding evidence from the early studies of DuPont, where DuPont had done radio-labeled glyphosate, and then they found the label in these proteins, you know, they gave it to the animal, and then when they got the tissues with the protein in it, they had the label in there, but they didn't see the glyphosate. Because the really awful thing is that when it's in the protein, it doesn't show the signal. You have to break the protein down before you can see it, and the protein's really hard to break down because there's glyphosate in it. So you have to use a lot of pepsin and trypsin, all these digestive enzymes. And he said DuPont had to run it for a week with these digestive enzymes before they could dig out the signal. And then they said, oh my God, the glyphosate's there. So the obvious thing is that it's in the protein, you know, it's in the protein. To me, it seems very clear. Time. Jen is worried because she's supposed to go on at 12:15 and we're supposed to have a break, so you're going to have a really short break.
Yeah, I just
we better quit.
Rodeo is Roundup to be used for aquatic weeds. It's approved to be used for aquatic weeds. So if you're wondering if it's in the water, yeah, it can be.
Right.
And then are your slides available on your website?
Oh, yes, right. They are on my website. They're already posted. I should have said that. You Google my name, but if you want to know, PEOPLE, people.csail, my lab, csail.mit.edu/Seneff, SENEFF. But if you just Google Seneff, you'll be able to find my website. And I've got them posted under a whole section on Autism 1 with all my Autism 1 talks. Okay, we got to quit. So thank you.